In this video, I will talk about studies that describe two fasting strategies that can improve the function of intestinal stem cells. But first, a short introduction. The basic structural unit of the gut is called the crypt. The lining of the crypt in the small intestine is composed of specialized cells, each with distinct functions. These include the absorption of nutrients, the secretion of gut hormones, the production of mucus and antimicrobial peptides, and the formation of tight junctions to ensure barrier control. The cells of the gut lining have a short lifespan of about 3 to 5 days and need to be constantly replaced by the new ones derived from intestinal stem cells. Any disruption in the replenishment of the gut lining can lead to malabsorption of nutrients, leaky gut, or inflammation. It was Hippocrates who famously stated that all disease begins in the gut. It would not be surprising if the exhaustion of intestinal stem cells as we age was the primary driver of age-related health decline. Is there something we can do to stave off the effects of aging on intestinal stem cells? It turns out there is. A study published in 2018 reported that a 24-hour fast improved the activity of intestinal stem cells in the aged mice. The mechanism of this fast-induced stem cell activity boost was the switch from glucose to fatty acid oxidation. The master regulator gene responsible for this switch is PPAR-delta. This receptor senses the presence of free fatty acids, and because fasting increases free fatty acids due to increased lipolysis, fasting was able to activate this gene, yielding the enhanced ability of intestinal stem cells to replenish the gut lining in the aged mice. Then, another group of scientists found an interesting detail. It turns out that intestinal stem cells are not all the same. The majority of intestinal stem cells are called crypt-based columnar stem cells, or CBC stem cells. They are more numerous. There are about 12 to 16 of them per crypt. They are continuously dividing and play a role in routine intestinal maintenance. But they can be killed or injured by toxins, chemo, or radiation. In contrast, a rare pool of cells called dormant stem cells are non-dividing and therefore resistant to chemo or radiation. There are only 1 or 2 of these cells per crypt under normal conditions. They are reserved for special circumstances and can play a role in intestinal lining restoration when CBC stem cells have been exhausted or destroyed. Researchers found that what makes these special dormant stem cells stay dormant is the presence of a signaling inhibitor called PTEN. In order for these cells to get activated, the removal of PTEN inhibition is needed, and this could be achieved in mice with a 48-hour fast. The authors of the study wrote, "Acute nutrient deprivation leads to transient PTEN phosphorylation within d-ISCs and a corresponding increase in their number. This release of PTEN inhibition renders d-ISCs functionally poised to contribute to the regenerative response during refeeding." It is important to stress that it is fasting that makes dormant stem cells ready for activation, but it is refeeding that activates them and makes them replenish the specialized cells of the gut lining. Both fasting and refeeding, not just fasting, are needed. All this research was done in mice, but it would be reasonable to assume that the outcomes could apply to humans. Hence, we could consider having 2 different fasting strategies to improve our gut health. A periodic 24-hour fast could be great for enhancing regular gut maintenance by CBC stem cells as we age, whereas a longer fast for 2 days, followed by a 5-day refeeding, might be needed in situations when the regular stem cell compartment is damaged or depleted, and one needs to tap into the dormant stem cell reserve.